All right. We got a few people on. What is this? I'll share a screen. I still got to get set up. We were a little slow getting in here. So y'all give me a second.
Yeah, but it's on top of something.
Yeah, don't forget to get a wet wipe, clean your desk. Okay, and then I need, it's like 10,000 things to open before class even starts. And it's ridiculous. I can't see everything. Every time I try to click on something, something else comes up. I don't know if I did that right. Okay. This one. This one. We're going to try with the Smart Notebook. I don't think I've done this in this class yet with the Smart Notebook. But I think it's going to be better for me to draw on this way. All right, so I could put up attendance. I could start attendance. Okay. So go ahead and do attendance if you can. And I am going to try and exit out of this. Okay. Now, why did my bottom thing go crazy? Who knows? Okay, so I can minimize this. Oh, I do need this. And I can close this. Okay, do we have anybody? Oh yeah, I was gonna open the chat on my computer. That seemed to work pretty well last time, so that I can keep my board clean. Which I didn't know you could have like multiple, you know, like as a user, I can have multiple devices zoomed into the same meeting, which is pretty cool. Oh, and my computer doesn't want to get on the internet. It's not one technology thing, it's another. Yeah, that's what mine's doing. That's weird. Well, it's not just you.
So it's not just me.
It's not just you. Well, now it says I'm connected, but now I can't get on. Try this again. Okay, let's see. And y'all can see what I can see, right? Oh. Yeah, it doesn't want to connect to the internet for me. Well, I can put the chat on up here until it works out. Okay. Yeah, I don't want to. Oh yes, Labster is due today. If you don't have Lab, you have Labster due. That's very true. Thank you, Hannah. Thank you, Hannah. She's going to keep us all in check. Do you have internet yet? Me neither. It's not just you. It's a good thing that this thing's hooked up to a wire. Okay. So let's go ahead and we're going to start. So I want to tell you a few things real quick before we start. What numbers of post-lab do we do? If you were in my section A, I sent you like pretend data because the spec wasn't working. Jennifer, if you have questions about specific ones, you can send me an email. I don't know if I'm going to get to it before I have lab today. I'm not going to talk about lab questions in class, but if you have questions, you can send me an email. I'm happy to answer it. but I don't know when today because I have lab this afternoon. So be patient, is what I'm saying. Okay, so before we start, I want to talk about our calendar. So if you go to our calendar right here under the syllabus, all right, this is what I want to look at. So I need to zoom out a little bit. It's too big. Okay, so we are here, right? We're here. No, we're Friday. I don't even know what day of the week it is. All right, we're Friday. We're starting chapter 5, section 1 today. So I took a look at what happened with the hurricanes and all that kind of stuff. So our exam 2 is still going to be scheduled for what it was normally scheduled for, October 2nd. I went and looked at our next exam. Our next exam was going to be on Wednesday, October 19th. No, October, what is that, 15? No, that's 19, 20, 21. Yeah, 21, the same day as drop day. But I went ahead and scheduled it one class period before that. So it'll be on Monday the 19th instead. So, and then I kind of have a plan generally through the rest of the semester. We're not going to get as really far as I wanted, but we got to roll with coronavirus, right? We're just going to roll with it. So if anybody notices an error, please let me know, but this is the updated calendar. Okay, so we got that taken care of. And I will have your last exam graded before your new exam. You have to take the next exam. So I know it's taken me a little while, but I'm doing the best I can. All right, so did y'all enjoy section 1? I love section 1. I absolutely love section 1. Section 1, 2020 has been a ride. Yes, it has. It absolutely has. By the way, do you have to? No, there are no Saturday classes for me. I shouldn't say that too loud with the door open. Nope. Okay. Remember we made up our extra day with an extra lecture over the weekend. So we're okay. It's fine. Okay. You're welcome. Okay, so chapter 5. Chapter 5 is super interesting. I love chapter 5 because it's so practical. And everything that you're going to see in chapter 5, when you experience it in the lab, you're like, oh, I read about that. Oh, I read about that, right? So this chapter is super applicable. I mean, like, unbelievably. It brought back so many memories for me being in lab and the things that I did and the problems that I ran into. So what I tried to do for our iClicker questions, there are some like regular iClicker questions in here, but there are some where I've made you the researcher and I'm asking you questions about what you would do in certain situations. So they're a little more technical than some of our clicker questions have been, but we're going to go ahead and try them and see what we can do with it. So before we get started, does anybody else have any other questions? Do we have any 4.3 iClicker questions? We do not, Caitlin, because we didn't get that far. We do not. We only got through 4.2. Yep. Okay. All right, so here we go. All right, so we talked in chapter 1 about dialysis, right? And you have your protein at some point during purification, and a lot of times you have it in a really high salt concentration, and so you put it into dialysis tubing, and then you put it into a buffer, right? So that you can exchange those ions. So this is an application of that concept. So we know that dialysis, I'm going to stop this one and I'll start again. Dialysis is frequently used when people have kidney malfunction or no function, right? And the whole point of dialysis is to remove waste products from the body. So based on size, what products do you think will not be filtered out through dialysis? So remember, it's blood. You take the blood out of the person, right? It comes out of one arm, goes into the machine. They clean it, and it comes back into the other arm, or the same arm, depends. But it's all about cleaning the blood. So what will not be filtered out? Ross, I know most people don't have to do dialysis, but most of y'all have taken a biology class and have probably heard about dialysis, maybe. I know if you took my biology 2 class, we talked about it. All right, so I give y'all till minute 30, so 5 seconds. We had 56 people. How do we only have 52 responses? Like, how does that work? Y'all. Okay, so here we go. So what's going to be filtered out? What is glucose? What's the formula for glucose? So are we not going to talk about, well, we're not going to cover 4.3 as much. No, we're not, Allie. I mean, there's only so many hours in a day. So unless y'all want to come to class on the weekend, but there's only so many hours in a day. All right, so glucose is a sugar. Anybody remember the formula? C6H12O6, right? Protein, what do we think of when we think of protein? Right, all of our amino acids, are we thinking something really small or really big? This is going to be something really big, right? It depends on the protein, but generally pretty big. Ammonia, what's our formula for ammonia? And come on, there's a chat. You could type it in the chat. NH4. Well, ammonia and ammonium are 2 different things, right? So which one's which? So what is ammonia? NH4 or NH3? We got 2 votes. NH3 is ammonia. NH4 is ammonium. Okay. And then oxygen, how does oxygen come? As O2. So these things are small enough that we have one formula for them, right? They can get through smaller membrane sizes, right? So the only thing that's really not going to be filtered out is what's really big. So it's really the protein that's not going to be filtered out. What answer choice was that? B. So what did y'all say? Y'all said D. What was D? D's oxygen. Well, you have to think about, in dialysis, you want to remove all of the waste products, right? So are you able to selectively pull out carbon dioxide versus oxygen? No. You actually have to pull it all out and then put back what you want. So Hannah, yes, you would put back in oxygen. You could put back in oxygen, but the patient can do that. The patient can breathe. They just can't get rid of ammonia waste products and things like that because their kidneys are malfunctioning. Their respiratory system is good, so they can still oxygenate their blood. They just can't get rid of waste products. So does that make sense? We did a lab with dialysis tubing. Yes, yes, yes. Not gonna make it. Good, good. Dialysis tubing is pretty cool stuff because you can buy dialysis tubing with different pore sizes. So you can buy the tubing that fits what you want to exclude or what you want to include, depending on how you want to look at it. Okay. So, all right, now we're getting to a little bit more scientific, right? Okay, a mixture of blue dextran, cytochrome C, and DNP glycine is placed in a Cephet X size exclusion column. Using the table below, what is the elution order of the colors eluted from the column? So you're going to put these 4 substances in a size exclusion column. What order will they come out in? Give y'all a minute and a half. still don't have internet. It's so weird. Gracie, you got internet? It's just me though.
I had to turn mine off and back on.
Oh, that's my deal. I'm on a hard wire on Ross. I'm on a hard wire on the computer in the classroom, and I'm trying to connect to my laptop. That's what I'm trying to do. Okay, so we're past our point of what I said I was going to give y'all. So, okay, so let's see. What did everybody say? Everybody wanted to say D. Why did y'all say D? If you do D, right, that's telling me that we're going to elute out small things first and big things last. That's what y'all said, first to last. Tell me about size exclusion. When you put these beads in this column, right? When I say a bead, I think most of y'all are thinking like a marble, right? A solid bead. These beads are not solid. They're like a meshwork. They're like, you know what it makes me kind of think of? It makes me think of a sponge, right? You know how a sponge has all these little holes and the water can go into the holes and stuff like that? Beads are the same way in size exclusion. They have all these holes. Well, if they have all these holes, if you're small, you're going to end up having to go through all the holes in all the beads. If you're big, you bypass them and you just go on the outside of the beads. So what's going to end up going faster? So if y'all watch the video, you remember I equated this to an obstacle course, right? So say there were two of us running this obstacle course. I'm gonna see 10 obstacles, and I'm gonna have to go through 10 obstacles. But the person next to me, Gracie, over here next to me, has decided, she just came to class, so I just decided to pick on her. She has decided that it's gonna be easier to get to the finish line if I just run along the side of the obstacles and not go through them. And that's exactly what happens with those larger particles. They go speeding through, right? So what should your answer have been? Your answer should have been B. Your answer should have been B. So y'all had it mixed up. You were thinking the small things are going to go through fast because they're small, but it's the complete opposite. Yep, so the blue dextran is going to go through the quickest because it's the biggest. I mean, that's huge. It is just going to zip straight down. And the smaller the molecule is, the more it's going to take a meandering path from start to finish. So it's going to take longer. Okay? Everybody okay on that one? Did y'all watch the video? Yes. Okay. Yes. All right. Good. All right. Based on the structure of Kamasi Brilliant Blue, some of y'all use this and some of y'all will use it today, right? Based on the structure of this dye, it's called Kamasi Brilliant Blue, which amino acids would react with this chemical? So you have to stop and you have to think about the structure of these amino acids. Try not to look at your notes. They're accessible, but try to answer without looking at your notes. I'm going to get a pen and check off the ones we did. So I give y'all about a minute and a half to kind of think about it. You really kind of got to think about this one. And there might be a reason why I boxed in some things on that figure. Okay, got 5 seconds. Okay, so what did you need to know about these amino acids in order to answer this? You have to know something about their what? Their structure. That's exactly right. Their structure, because their structure drives function. So if we look, this one's methionine, then we have serine, then we have tryptophan, and then we have lysine. So I went ahead and put them in for you. So what do you notice about one of these amino acids? There's something particular about one of these amino acids. What did we pick? Whoa, we got quite a spread there. One of these amino acids has something that kind of complements our dye. They know what it is? Not matches, but complements. Are you all awake at home? Oh, well, tryptophan has rings, has aromatic rings, sort of like the structure. Y'all are not awake. Lysine is the only one with a positively charged side chain. What do you notice about Kamasi? So what do you think is going to happen? This negatively charged residue over here is going to interact over here. Positive and negatives attract. Yes, good, good, good. So what should we have said? We should have said that it was lysine. That's D. I keep like looking and then having to look again. D, right? Because lysine has that opposite charge, right? So this one's going to react with the chemical. So it will really react with any charged amino acid. And so all proteins are going to have some kind of charged residue. So Kamasi will work for any protein. Some better than others, but it'll still work for any protein. Okay, like I'm super excited about this question. I had so much fun with this one. All right, you have had a long and difficult semester. I know. And you've been doing research and it's been so challenging, right? And you have almost purified your protein of interest. And I'm going to call this protein of interest YOPI, right? Your protein of interest, YOPI, okay? So this is your YOPI protein. But when you stained your gel electrophoresis with Kamasi, we have 3 proteins. When I look at those 3 proteins with a mass spectrophotomic analysis, right? If I analyze each of those 3 bands, I learn the size and I learn the isoelectric focusing point of all 3 of my contaminating proteins and my protein, okay? So I know I have a mixture of 4 proteins. I only want YOPI. I only want my protein of interest, right? I guess that's better than MOPI, right? YOPI.